thank you so much for having me, and thank you for having me. I really appreciate you guys inviting me. I've admired lots of papers from your guys' group, read them often, and so it's nice to tell you, for a chance for me to tell you about what we've been working on in our group. And as you can see, what I'm going to talk about is really a collective effort of many, many people. It kind of started with my former postdoc, Charles Fisher, my good friend, who now runs a artificial intelligence company. A lot of it was, worked out with my former student Wenping, who's now at KITP, my former postdoc Bobby Marsland, who actually left science to go study to become a priest, and Alvaro Sanchez and Josh Goldford were very involved in trying to take this theory and relate it to experiment. And we had a lot of fun working on all these things together. And as you can see, there's a series of papers that have been produced. So as I said, please don't be afraid to ask me questions or interrupt. And so let me start with some very basic statements, which is that, even though I am in a physics department, I consider myself a physicist. Biology is clearly very different from physics. It's generally made out of heterogeneous components. It functions out of equilibrium. It's, as John Hoffield likes to emphasize, matter with function, and it's a historical product of evolution. And so the question becomes, given that all these differences between living matter and non-living matter, how do we do theoretical physics? And how do we use physics principles to understand these kinds of biological systems? And I would argue the reason that this is especially difficult is that we can't actually rely on many of the pillars of modern theoretical physics. So I would say the great successes of 20th century physics have been basically, centered around a couple of ideas. The first is of symmetry. It can be the symmetry group of a lattice in condensed matter physics or solid state physics, or it might be symmetry of gauge theory. So here you have eightfold way of QCD. But biological systems, by definition, except for some very specific settings, largely don't have these large amounts of symmetry that we associate with most of the successes of theoretical physics. The second thing is that biology, of course, works out of equilibrium. And so all these energy functionals we're used to, Hamiltonians, free energies, things like that, they're really nowhere to be found in any obvious ways. And so I think this kind of challenge of modeling these systems that seem to have lots of structure and order, but without having access to these kind of central concepts of physics, really mean that we have to do something kind of very different. And I find that to be a really exciting proposition as a theorist. So I guess one of the things, if you don't take away anything else from this talk, is that I want to ask, how do we do theoretical physics, or maybe it's much closer to theoretical biology, without symmetry, forces, and energy functionals? And I think there's going to be, I'm going to try to tell you one approach that I think is quite promising and that we've been, actually using in a wide variety of systems, well beyond the microbial communities I'm going to tell you about, which is kind of to exploit the deep relationship between randomness and complexity. So I think one of the defining features of biological systems is that they're quite complex. And for whatever reason, we know historically, even from a strain of physics, that complexity somehow can be well captured by randomness. So, just historically, I think the inspiration for this kind of idea comes from Wigner, who after World War II was thinking about how to model uranium, right? And the interesting thing about uranium is that it's a very, very complicated nuclei. We all know it has 92 protons, 146 neutrons, 92 electrons, and there's no way we could solve the Schrodinger equation for such a complicated, system. And so Wigner had this crazy idea where he thought, instead of thinking about the actual Hamiltonian uranium, I should just replace this by a random symmetric matrix. And it has to be symmetric because that's the constraint that comes from unitarity. And so he basically took this complicated atom and replaced it with a matrix that looks like this on the right-hand side of this equal sign. And so the question becomes, what's surprising about this is that by doing this, he was actually able to reproduce many kind of observed experiment. And the most famous of these was, at the time in the 50s and 60s, people were very interested in the nuclear structure of uranium. So they were doing scattering experiments where they would basically bombard uranium with particles and measure the scattering cross sections. And you, of course, got resonances, which correspond to the peaks on this plot. And if you looked at the distance between these peaks, you saw that this distance between the peaks actually followed what now is known as the Wigner surmise, which is the same thing as that could be described by a random symmetric matrix. So even though uranium is this very structured, complex system, it turned out that many properties of uranium could be actually captured by just thinking about randomness. But the important point was that you had to put this kind of symmetry constraint in, because that was where the physics came in. And so the reason we know that this works now is that the statistical properties of these kind of eigenvalues of these random matrices become what we know as universal in the thermodynamic limit, meaning that if I have different realizations, eventually, as these matrices get very big, the kind of eigenvalue spectrum kind of converges to the same spectrum, and the fluctuations between different realizations disappear. And for this reason, one random matrix or one complex atom, as far as eigenvalues are concerned, are indistinguishable from every other atom. So
Pankaj, just to interrupt for a second, so these are the energy levels of the electrons, or what are these eigenvalues representing really?
Of the nucleus.
The nucleus.
It's a nuclear shell model.
Okay.
Of the resonances of the nucleus. It's all nuclear. Cool. The electrons don't play any role. It's protons and neutrons. Okay. So you have the proton, neutrons, and nucleus, and you scatter off the nucleus. And these are basically the resonances of the nucleus. These are the energy levels of the nuclear model. And so what happens is that the eigenvalues are the eigenstates of this nuclear Hamiltonian. And the interesting thing is, of course, that the distance between these things follows a universal distribution that, even though uranium is clearly not random, is well described by this randomness. So this kind of hinted at, this was the first hint that if something gets complicated enough, maybe we should model it with a random system. And of course, about 20 years later, this kind of ideas were brought into ecology by Robert May, who was in your side of the Atlantic, who basically thought of thinking about ecosystems using large random matrices. But of course, what he was really thinking about was a large linear random dynamical system. And he did this, and he inspired a large series of papers, which were called the stability and complexity debates, based on these kind of analysis of random systems. But for many reasons that will become more clear, I think the problem with May's approach, even though it was so influential and successful, was that he didn't think enough about the ecological and biological constraints that you have to put in the random matrices you draw from. And in particular, it's true that what I'm going to try to argue to you is that properties of complex biological systems indeed should be well described by appropriately chosen random systems. But the caveat is that we have to put enough of the ecology and biology in to where we draw the random systems, and these kind of manifest themselves as constraints. So you have to really take random matrices that reflect the ecological and biological constraints of the system we're studying. And so I know this is very abstract, but this is going to become very clear because this is basically the high-level picture of what we're doing, and I'll show you how we're adapting this to really think about microbial communities. So the basic idea, what I want to tell you for the rest of the talk, is how we're trying to adapt these ideas and extend these ideas of equating complexity as randomness plus constraints in the context of microbial ecosystems. And if this was a different talk, I'd show you how these same ideas manifest themselves in other biological systems. But in order to do that, we have to really think about how the ecology of these microbial communities function. What are the minimal ingredients you have to put in a model that captures enough of the ecology so that we can really say something about real biological systems? So the important thing that I want to point out is that ecology, and I think this is well understood by almost every microbial ecologist, is that microbial ecology is shaped by cellular metabolism. And what's really important about microbes that makes them very different from many of the classical ecological systems is that they don't consume other species, but instead consume and produce small molecules. So for our purposes, the way we think about a microbe is that it's an organism that eats different resources. It takes some of that energy and uses it to grow, and it takes the rest of the resources and leaks them out as metabolic byproducts. And so what's interesting about microbial ecosystems is even if you put an external environment in, or you put a single resource in, through cross-feeding or this metabolic byproduct production, microbes will fundamentally change their environment. And so what's interesting is you put a single resource in, microbes produce other resources, which are then in turn consumed by other microbes, and then which secrete other resources, and so forth and so forth. So you get this complex web of resources and consumption that basically set up these things. So, of course, what's important is the process of how much energy you use from these resources, and what gets excreted in response to what gets consumed is basically the study of metabolism. And as this picture shows you, and this is a picture I've stolen from the KEGG database that is actually quite zoomed out, metabolic networks are super complicated. We actually know many of the enzymes that go on. This is the TCA cycle, this purple cycle in the below, but it's just super complicated process. And so the question is, how do we kind of incorporate this into our models? And so the basic thinking that we had was basically to draw inspiration from Wigner and to think about how do we model this complexity. And just like Wigner said, oh, we could forget about all these uranium atoms and think about all the complexity and replace it by a random symmetric matrix. What we're going to do is we're going to basically model the complexity of consumer preferences, meaning what resources each microbe likes to eat, using a random matrix, basically random consumption. So every microbe is defined by what resources it likes to consume, and we basically choose that to be a random matrix. And we're also going to model metabolism, this complicated metabolic network, by another random matrix, which we call random metabolism, which tells you that if I consume resource alpha, how much of resource beta do I produce? Right? So it's essentially a stoichiometric metabolic matrix. And so what we're going to do is we're going to just think about these kind of systems using what we call the microbial consumer resource model, because it basically builds on a very long tradition in ecology of consumer resource models, where basically what happens is that we can think about the abundance of species I, which is given by Ni. And the idea is that these species grow proportional to this growth flux, which is how much energy they consume. But when they consume things, not only do they consume things, but they also leak out other resources. So that's this J out. So they take in J in of metabolites, they leak out J out. And the important point is that what they take in is basically given by a random matrix, Ci alpha, which is the consumer matrix, times the abundance of the resource in the environment. And what they leak out is basically proportional to some leakage rate that tells you what fraction of the resources are leaked out, back out into the environment, times this random matrix dB alpha, which is like a random Dirichlet matrix, which basically tells you the fraction of energy that's in resource alpha that gets excreted in resource beta. All right. And so we've taken all this complexity and we've basically subsumed it in these two random matrices. And what I should point out, and what I really want to point out, is that this is quite different from what May was doing, because we've actually put in a minimal amount of structure that was missing in May. So we basically have made a cartoon, the minimum cartoon model of these kind of very simple pictures. And so the constraint we've imposed, whereas May imposed no constraints on the random matrices, what we've imposed on the system is that we've required that this kind of complex dynamics reach some steady state. So what we're going to look for is steady state solutions of this kind of complex dynamics of resources and species. And so what?
Just to ask, so have you randomly imposed a specific steady state? Is that what you're saying?
No, I haven't. I've chosen random C and D, and then what I'm going to do is look for solutions where dni dt and dr alpha t equals 0.
Okay, so yeah, but you haven't chosen the steady state, but
No.
you've randomly generated some numbers, and they specify a steady state, and you're going to find it.
Yes, and what I think is important is that these are structured equations. They're not just dni dt equals some random matrix times ni. There's a lot more structure in these equations. And so it's like working in a different ensemble than what many ecologists will work in. I think that's what's important. There's like basically a flux. What I want to point out, what I think another way of saying it is what the second equation is a flux balance constraint, right? So the system I choose the random matrices, and the system self-organizes to a steady state that balances fluxes.
Okay.
And that's the properties I'm interested in.
Just if also another question. So on the previous slide, I think you sort of emphasized that these microbes, they don't feed on each other, but they secrete, and then the other bacteria can take it up, or microbes. So why did you stress that? I mean, it could be included in the model, but it's just a different type of model. Or was there a particular reason why you stressed that they don't feed on each other? I mean, you could have microbes like amoeba. I guess they're also called microbes. Yeah. They could feed on by phagocytosis on other bacteria or something.
I agree. What I want to stress is that what I think what I should have said is this model does not capture those kinds of interactions, but instead assumes that competition happens through competition for resources, right?
Sure.
And that is due to empirically, I think that's a very large and dominant form of organization of microbial communities. Though, of course, there are all systems where
to make some assumptions. But when you say also on the next slide again with the steady states, I mean, do you run into a problem of multi-stability, or do you check for that, or how do you deal with that problem?
No, there isn't. And I left that whole part of the talk. So in the process of doing this, when we simulate this, we actually, one of the things, we have a line of papers, a couple of papers where we show that all these consumer resource models can be mapped to a constrained optimization problem in a literal sense. And so it turns out, for reasons, if you just have consumer resource kind of things like this, we do not ever run into multi-stability. It has to do with this kind of mapping to constrained optimization. You can show these things are semi-convex. For these things with cross-feeding, I've found, I know there's a bunch of papers discussing whether you can ever get multi-stability or chaos in these consumer resource models. And there's a series of papers from Guy Bunin and Giulia Baroli saying that if I allow the kind of interactions you were talking about, alpha ij in species-only models, you can get these. But when I run things for those kind of parameters where you see that kind of dynamics, I often find that there's some resource that becomes negative, so it's an unphysical solution. They claim otherwise. We haven't, because of the pandemic, we haven't had a chance to sit down next to each other and work it out. But I would say generically, it's very hard to get in these models. And it has something to do with constrained optimization and the fact that we're very carefully imposing energy conservation. So one of the things we're very careful about, and I know, Robert, in this last paper you thought a lot about this too, is that we're very careful about imposing energy conservation, that there's no energy created in this cascade of metabolites coming through. And my feeling is that in order to get these runaway solutions, you need to violate that implicitly, even though you think you're not. But you can't disagree.
You can't choose some nonlinear function. I guess R you have there, right? C is the external nutrient. R is some function of uptake and
oh yeah, no. You can.
No,
so R. Sorry, a lot of frustration. Yeah, if I make it look like antiferromagnetic coupling, so I put a lot of frustration I certainly can. I very much do mean the linearized dynamics. Thanks for pointing that out. No.
Yeah,
if I make the J in to be a sufficiently nonlinear function with high Hill coefficients and very different things, you can get chaos. Absolutely, you can.
Thanks.
I'm focusing much more on linearized dynamics, but you have to make it quite nonlinear to do that. And even then, I haven't actually played with nonlinearity and trying to think about whether energy conservation is balanced. I haven't thought about that. Right.
So how do you impose an energy conservation?
It's not in
the energy coming in here. I mean, yeah.
I left him out of these slides because I didn't want to make the formulas very crowded, but basically each resource is associated with an energy, W alpha. And so all these kind of, stoichiometric metabolic matrices are about what fraction of energy ends up in one thing to another thing. So we're ensured that you never, basically leak energy. So basically the amount of energy in this out is the amount of energy in this in minus grow. You can just do that very easily by just the structure of the equations.
Okay.
And then dB alpha is basically the columns. If I sum over a column, it sums to 1. So it's a Dirichlet matrix.
Okay.
So quick question. So you also have the MI term, which is the death term, is it?
Yes.
So when they die, they give the energy back out in what form? In all J in or?
No, when
you have to redistribute them.
No, that's leaked out of the ecosystem. Thanks. I didn't talk about that, but that's leaked out of the ecosystem. And at steady state, what basically happens is that the death term exactly equals the energy conservation term, right? How much you grow. And so you can just balance energy that way. So there's many ways to balance energy in this thing.
Okay, so you're constantly leaking energy, but then so you've got one external resource.
No, you're consuming
it. Yeah, but you have one constant. I mean, you have to put energy in, and that's your external resource.
Yes.
And that is in terms of that's in the graph. You've got just one external resource, and then they produce a lot of different resources. So, yes. But in your model, you've got a bunch of external resources for all eyes.
Oh no, because right. So there's one external resource which I'll call 0, right. The one that's supplied, right? So R0 alpha is 0 for everything except for the externally supplied resource.
Okay. Thank you.
That's how I impose it. And then the point is the amount of energy you're flowing in has to equal the amount that's lost due to dilution, which is just this term, this tau inverse R alpha term, plus the amount you consume to grow, which is exactly equal to the death rate at steady state, because it's like a chemostat.
Okay. Thank you.
Hey, there was a question on the chat box, and we talked about this already a bit, but do all non-equilibrium solutions necessarily violate energy conservation? I suppose not, right? Everything is balanced. No. But the question was also, are there scenarios chaos is realistic?
I think Guy and Giulio and those guys would certainly claim so. I would just say they focus more on lockable Volterra models. I make very consumer environment specific models because I feel like that's what you can manipulate and experiment very easily, as I'll show you, the environment. I would say no, but they're very confident yes, and they're very smart people. So like I said, we haven't resolved it. Like I feel like you always implicitly violate energy conservation. If you have consumer only reactions, the question then becomes, and of course, if you have lockable Volterra, you have many non-energy conserving interactions, so their stuff can happen. The question becomes, if I start with a consumer resource model and I put in a small amount of non-consumer mediated interactions, can you get chaos or not? I think that's the domain where we have disagreements. It's certainly true in general. Okay, you can get chaos.
Thanks.
All right. So the important point I want to next go on to make is that we can choose these matrices. We have freedom in what ensemble we choose these matrices from. And in particular, we can, put different amounts of structure into what we do. So for example, we can imagine that we have completely unstructured metabolism and consumer preferences. This is the top row where we see that like everything kind of looks random. Alternatively, we can put, tiers, recognize that metabolism actually has different kinds of byproducts and resource types, and maybe certain families specialize in certain kinds of metabolites. For example, in enterobacteria, we don't eat sugars. And we can basically put, for example, block structured kind of things. And I didn't, put the slide in, but what I want to tell you is that we actually wrote a Python package. It's called the Community Simulator. It's a PLOS One paper. We use it to simulate everything. It makes all this super easy to simulate. You can do everything in 3 lines, all the simulations I'm going to show you. And we use this trick where we map things to convex optimization. So you get around the fact that these ODs are stiff. So it allows us to do large scale simulation in a way that I think would be very hard to do if we directly integrated these things. So I encourage you to look at this package if you find this interesting. And all these kind of simple structure, whether you use structureless thing or whether you put in these kind of tiered secretion models, that's all built in. It's just parameters, and it's really easy to play around with all this stuff. So, but the point is, the larger point I want to make is that you can make the assumptions and ask, what assumptions do I have to make about metabolism and consumer preferences in order to reproduce these kind of things? So I hear an echo now, but so, you know, after we kind of wrote down some of these models, the first real test we had of this was actually published in this science paper by Josh, who's like really brilliant, and with my friend and collaborator Alvaro Sanchez. And what we did, what Alvaro had, he had this amazing experimental system where he just went out to his backyard and he basically got a bunch of bacterial communities from leaves and plants and soil. And what we did is that we kind of grew them in what we call these minimal synthetic environments, which were these kind of like M9 media, which is like 9 ingredient media with a single carbon source. And we asked, what we did is we just kind of grow them up for 48 hours, dilute them, and keep on doing this over and over and over again. And we did a bunch of experiments. You reproducibly went to a steady state. There was a bunch of things you could see. But the main thing I want you to take away, this is a very busy slide, is the top is basically real experiments. The bottom is basically using a very simple tiered consumer resource model. And we could reproduce many, almost everything that we observed, much to the surprise of Alvaro and Josh, because Josh, we'd get a new piece of data and Alvaro was convinced that that the model wouldn't reproduce it. But we used the model to design the experiments, and the model actually reproduced almost everything we did see in the experiment. So this was kind of very encouraging that this kind of very dumb random matrix theory approach could actually reproduce almost all the qualitative patterns we saw in these kind of experiments. And of course, I couldn't tell you about what each species is doing because that's not what the model is designed to do, but at the level of an ensemble, at the level of ecosystem level properties, we could actually just see that cross-feeding was the dominant thing shaping these kind of simple experiments, and it could reproduce almost everything we could measure qualitatively using 16S sequencing. And so after this paper, we got interested in asking, well, this is a kind of contrived setting. Can we say something about, like, you know, natural patterns? And so what we did after that with Bobby is that we kind of started looking for patterns in across labs, across experiments. And so one of the big projects that had been going on at the time was this thing called the Earth Microbiome Project. That's what EMP stands for. So this was this kind of crazy project where they measured microbial communities across, you know, thousands of communities. They kind of measured what was there. They did this large scale survey. It's quite impressive. There's, you know, animal associated communities, there's ocean communities, there's soil communities. So at the end, when they had to publish their Nature paper or whatever it is, they had to come up with some plot, and the main plot they found, the pattern they wanted to emphasize, was this kind of nested pattern you see on the top. So this kind of figure on the top is experimental data. Each column corresponds to a different community, and each row corresponds to a different taxa, or you can think species. And if a taxa occurred in a community, what you see is that you see a colored dot. And you can ignore the colors. The colors just tell you what kind of community it is: soil, ocean, things like that, animal associated. And what you see is that the taxa that occur in the least diverse communities, in the least rich communities, also occur in the most diverse communities. So that's this kind of upper triangular pattern. And so what we did is that we just kind of simulated this using this microbial communities, and the only thing we changed across communities, so we drew one random matrix for C, one random matrix for D, and we just basically changed how harsh the environment is. So what's the natural death rate of these things? So every dot on the bottom are simulations where every point, all we did was sample this MI from a Gaussian distribution. And what you can see is that you can easily reproduce this upper triangular pattern. And what it indicates is that actually what's dominating what species occur in a community is the fact that species are limited by competition for resources. And so we had kind of reproduced this pattern, we sent it off to review, and a very smart reviewer who we're grateful for said, that's great, you can reproduce the pattern, tell me how I can break this pattern. That's the only way I'll learn something from it. So we spent a lot of time, it was actually very hard to break this pattern, but we finally figured out that if what limited what species were in a community was not competition for resources, but instead this kind of immigration to different communities. So we basically had this dispersal limited. So basically you had very few species that could colonize a place, then this kind of nested pattern disappeared. So what we learned from this is that this kind of nested pattern is an indication that what's shaping community structure, what species are present, is actually competition for species rather than immigration or dispersal. So this was just one example, and we managed to do this with many, many things. So these are a series of experiments from Otto Cordero's lab, where he basically took ocean communities, grew them on these kind of beads that were seeded with a single carbon source, agarose, alginate, chitin, kerogen, and then what you have on the bottom are taxa. On the x-axis, you have time. And what you saw was this kind of succession dynamics where certain taxa appeared first, but they kind of disappeared and were replaced by other taxa. And just by doing kind of random matrices, we could reproduce that pattern. It occurred generically. Another important observation they had was that it seemed like if you grew something on a community on agarose, and then you grew it on alginate, and then you grew it on a bead that had both agarose and alginate, what happened was that the community on two sugars just looked like a linear combination of the community on a single sugar. And you can see that we could also reproduce that. And again, the reviewer asked us, when can you break this pattern? And what we found was that this kind of pattern of linear additivity basically corresponds to having a structured metabolism. So in particular, if we did these simulations with matrices that were completely unstructured, like this D up on the top, we couldn't produce the pattern. But as soon as we put any kind of block diagonal structure, this kind of linear additive structure reappeared. So this modularity seems to reflect the fact that metabolism is modular. And if you go to this scientific reports paper, you'll see many, many more examples where this kind of random consumer resource model was able to reproduce kind of all these experiments and patterns you see in lab experiments and large scale surveys everywhere. So we were quite emboldened by this. We were actually frankly surprised that this kind of dumb method could do all this, but it seemed to be continuously doing this. And so the next question we wanted to figure out is how do we understand what this microbial consumer resource model is teaching us a little bit better? And basically for 4 years, me and Bobby tried to solve this model analytically using the cavity method, which is a mean field method. And it literally took us 4 years to do it. But finally, a couple months ago over the summer, finally managed to work out all the kinks. And so what's surprising is, despite the fact this is a system of nonlinear equations with no energy functional whatsoever, no Lipyanov exponent, you can actually solve for a mean field theory of this. And just because I spent a lot of time deriving these equations, here's a big list of math that means nothing to anyone except for me. But the point is that it was a really complicated solution. You could derive these things. It works reasonably well with numerics. And that's fine. But what is the lesson this analytic calculation taught us? So what was interesting about us is that we wanted to understand how do you have, when you have these complicated communities, there's basically two big processes that are going on. The one is that bacteria and microbes are cooperating because they kind of are secreting these metabolic byproducts that are then eaten by other bacteria. On the other hand, we know that the bacteria are also competing for these resources. So you kind of have this cooperation through cross-feeding, but at the same time, you have competition through competition for common resources. And we wanted to understand what affects what. So it turns out that if you kind of do these analytic things, you can derive some analytic formulas. And the important thing we find is that actually the competition and the cross-feeding kind of are decoupled from each other. So if I ask about the average resource abundance, so this is basically the average amount of resource in the ecosystem, you can see it's just set by the amount of leakage, which is the amount of the fraction of resource that's leaked out as metabolic, but it doesn't depend really on the properties of this kind of consumer matrix. So this matrix C that tells you what each thing consumes can be basically characterized by a mean and a variance. And what you can see is that I can change the mean and variance of this consumer preference matrix, which is this color bar, and it doesn't really, and the amount of resource doesn't depend on that. Instead, it just depends on how much I'm leaking out into the environment. On the other hand, if I think about the biodiversity, so this basically tells you something about the species abundance of distribution. So this kind of quantity that I plot on the left is basically a proxy for what do the species abundances in a community look like. You can see that it's largely set by these kind of what species consume, but largely independent of the leakage fraction. And so you kind of have this funny self-organization where the cross-feeding is set by the where the amount of resources in the environment are set just by leakage, but largely seem to depend on what actually the species consume, and what species are present in the community, on the other hand, don't really care about how the resources are created, but instead just care about how much they're competing with these with other species. So you have this kind of very, very, very funny kind of decoupling between competition and cooperation. Even though both are occurring, they're kind of like decoupled processes. Once the resources are in the environment, the species don't actually care how they got there, whether they got there from cross-feeding or I supplied them externally. Then they just view the other microbes as competitors, even though the same microbes are leaking the resources out. So it's just like a kind of very funny self-organization that seems to be happening in these ecosystems.
Can I just ask something about your mean field theory? So
yeah,
of course you showed the ODE system, yeah, and that's a kind of a mean field theory already, yeah. It's a well-mixed system. You neglect fluctuations, but then somehow you mean something different here because you also calculate essentially variances. So is it something like a noise approximation, or what is it exactly?
It's literally a mean field theory. So you see that like, what it is that I treat every species as identical. So it's like a mean field theory, like in the way we mean in any disordered physics system. So what I'm calculating is averages over these random C's and D's, but also over different N's. So it's like a self-averaging system. And the reason I can do that is let's look at this, for example, J out term. You see this J out term is a sum over many, many things. So if I have many, many resources and species, right, this is a large sum. So I can think of this flux as having a mean and Gaussian fluctuations around that mean. And I can think about the growth rate for different species as having a mean growth rate over all species in the community, plus some Gaussian fluctuations. And so what I'm doing is I'm basically doing, what I mean by mean field theory is literally the mean field theory that you would use in replica or in any disordered spin system or any disordered physics system.
Okay, it's basically, yeah. Sorry, it's essentially a way to deal with these random matrices, yeah? So
it's a way to deal with these random matrices, and instead of telling you something about each abundance Ni, I can tell you about the statistical properties of these Ni's and R alphas. So I can tell you what's the average amount of R alpha, what's the variance of R alpha over all alpha, what's the mean of Ni over all species, what's the variance of Ni, what fraction of Ni are non-zero.
Okay.
Those are the kind of things I can ask. Those are the thermodynamic quantities. But I can't tell you about the abundance or species, the species abundance of a particular species or the resource abundance of a particular resource. I average over all those.
Okay.
So when I write this is the average amount of resource averaged over all possible resources in the ecosystem on the top here. And this is basically, you know, what is the distribution of species abundance in the ecosystem look like? So this is basically the variance over the mean squared of the species abundance distributions. And so this is very common in disordered physics system, a way of dealing with random matrices. So I think I only have like 5 minutes left, so I'll just tell you the last 5 minutes and then we can be done. So, you know, this raises the natural question of, okay, you have these kind of crazy real systems, and we seem to be able to capture at least quite a few properties using random matrices. So why do these healthy, diverse communities behave like random ecosystems? And so this is something we spent a lot of time thinking about. And the answer seems to be and to think about this, what we did was we imagined a thought experiment. So imagine that I started off with some community of specialists. So each species eats just a single resource. Then I add, and so, you know, the consumer matrix in this thing basically looks like, you know, a diagonal matrix with ones on it. And then what I started imagine doing is started adding a small random amount of off-target consumer preferences. So what I'm going to imagine is that my actual consumer resources are some, you know, preference for some resource with some small amounts of off-target consumption. So basically what I do is I say my consumer resource is kind of structured matrix plus a random matrix that's defined by some mean and some standard deviation, mu and sigma c. And importantly, because of we want to take good thermodynamic limits, we're going to scale the mean with the mean goes like mu over m, where m is the number of resources, and sigma c goes like square root of m. So we're basically scaling these in a thermodynamically consistent way. And we asked what happens as I increase the amount of off-target preferences. We know if there's no off-target preferences, every species will just eat its own resource and survive in the community because nothing competes with each other. But as I start increasing the amount of off-target consumption, you start increasing the amount of competition between species because they're competing for these off-target species. And so what happens actually is quite interesting, which is that as I increase the amount of randomness, so what I'm showing you in these kind of square matrices are the consumer preferences. So you see over here, you know, you only have, you know, basically diagonal matrices. You can barely see the noise. I start increasing the noise, and already by the time I get to over here, what you see is that many of the species have gone extinct. So that's the blue line. The blue line shows you what's going on in these kind of matrices I've shown you. And the red line shows what's going on for a completely random matrix where I don't have this kind of central diagonal part, the structured part. And what you can see is very quickly, even though it looks like by eye that it's clear that, for example, for sigma c equals 1 and m equals 100, that means 100 resources and a little bit of noise that looks like this, that it looks like I clearly have a very strong diagonal signal. The actual resource, the actual properties of the ecosystem, and what I'm showing you here is the fraction of surviving species. So up here, all species survive. Down here, about half the species survive. And what you can see is by adding noise, I basically become indistinguishable from a completely random ecosystem. And what's interesting about this is that as I increase the size of the ecosystem for the same level of noise, so this is sigma c equals 1 and 100 resources, here I have sigma c equals 1 and 500 resources, you see that even though I would say the bottom matrix everyone would call basically a diagonal matrix, it looks indistinguishable from something that looks completely random, the ecosystem level properties. And this is what we call a phase transition to typicality. The idea is that even a small amount of off-structured noise can dominate the structure just because there's so many different ways to be off-structure. So as things get more and more complicated, basically what structure you have gets masked by the noise, and there's a phase transition in a very formal sense to this typicality. And you can do it for all kinds of, numerically we can show this for all kinds of stuff, and analytically we can understand this using random matrix theory and this kind of cavity method we did before. And I think for the sake of time I probably won't tell you about it, but we have a bunch of analysis on how you can understand this kind of phase transition to typicality. And it's kind of, if you want to know more about it, it's in this Physical Review E paper by Wenping that we had a very hard time publishing, but I think is a pretty good paper. I'm pretty proud of it. So as I said, we can understand this phase transition to typicality using random matrix theory. It has something to do with the Marchenko-Pasteur distribution. And what I want to emphasize is that this kind of phase transition is actually I would say well studied in information theory for the last 15 years, and they're usually called information plus noise models. And the basic idea is that imagine I have some structured matrix that contains the information, and I add some noise to get a new matrix. The question is, when can I recover this information? And we know from these kind of information theoretic analysis, there's actually a phase transition from when you can recover the information completely to when you can't recover it at all. And maybe the most famous example of this is something called compressed sensing. So you can relate this to information theoretic ideas as well. So what is the implications of this typicality? Well, the implications is there's good news and there's bad news. The good news is that it seems like we can make predictions without knowing all the details. So if I have a complicated ecological system, it's going to be very hard to measure every interaction. But if there's enough noise in the system, which I would argue in biology there always is, if we want to know about macroscopic properties and large-scale patterns, we can actually just replace this with a random matrix, and it'll allow us to know about the ecosystem and make predictions and at least understand what processes might be driving some of these large-scale patterns, as I've shown you. The bad news, I would argue, is that it's going to be hard to have bottom-up engineering in large ecosystems. So there's a big effort in synthetic ecology, in many things, to kind of study 1-2 species ecosystems and try to build these up and engineer very large and complex and diverse communities. And I would say because of this kind of transition to typicality, where you lose the structure you put in because of off-target effects, this is going to actually be a very hard task. So I think that basically concludes what I wanted to tell you. So what I wanted to say is that the talk basically had 3 major claims. The first is that the large-scale reproducible patterns we see across microbiomes are emergent features of typical random ecosystems. So at least the patterns we've observed with 16S at the resolution we're looking at seem to be very consistent with the kind of patterns we can get from random matrices. The second thing is that this kind of transition to typicality suggests that these kind of diverse ecosystems will always, as long as they're healthy enough, and I can explain to you what I mean by healthy, will always behave like random ecosystems due to non-specific interactions. And the final thing I want to point out is that microbiomes kind of reflect this kind of very funny balance between competition and cooperation. Basically, the cross-feeding basically sets up the resource environment in which the microbes compete. And what's interesting is the microbes are both setting up the resources, but they're also competing simultaneously. And there seems to be this kind of decoupling, at least in diverse communities, between competition and cooperation that I find kind of interesting, and we saw from our analytic solutions. So finally, I'd like to thank, again, the people who did the work. This is Josh Goldford, Wenping, Bobby Marsland, and it was really a team effort, along with Alvaro, who really forced us to think about how we can relate this to experiment, and the NIH and the Simons Foundation for funding, for allowing me to think about these crazy ideas. So thank you.
Thank you so much, Pankaj. Fantastic talk. So yeah, so there's some time for a few questions. Can you hear me?
Yeah, I can hear you. Yeah, I can hear you.
So there's a bit of time for some questions. So either raise your hand and unmute yourself, or type them in the chat box, if you like. So in the meantime, wait, yes, Tom, you raised your hand.
Yeah. So on the slide where you said there's this transition as you increase the randomness, as you increase the noise, you get the random curve and the engineered curve converge to the same point.
Yes.
At 50% survival or something.
Yep.
And you said that there's a phase transition.
Yep.
And obviously with the statistical physics hat on, I would say, okay, what's behaving discontinuously or something in the thermodynamic limit there?
It's a great question. So I can show you. I have a graph. So there's 2 ways to think about it. From the random matrix theory perspective, what's happening is there's a spec. So the phase transitions can be thought of in 2 ways. Either a spectrum, right? An eigenvalue, there's a gap that closes to 0. That's the quantum phase transition way of thinking about it. Or there's a susceptibility that is discontinuous. And it turns out that both those things happen at that phase transition in a certain class of models. So in some models, it's a phase transition. In another class of models, it turns out to be a crossover. But you can see in the phys-rev, but basically what happens is the susceptibilities that appear in the cavity equations, which are basically ask what happens to the ecosystem if I make a small perturbation in the death rate of everyone, diverges. Alternatively, you could think of these consumer resource models as, maybe I'll just share this real quick. I'll just go back. Alternatively, the thing you can do is you can map these consumer resource models of this class of models without cross-feeding to an effective Lotka-Volterra model, right? And you have an effective interaction matrix, which is basically described by this C bar, which is basically the consumer preference times each other. So it's basically what's called the Wishart matrix. And what you can do is you can look at the eigenvalue spectrum of this kind of interaction matrix. And what happens is that that eigenvalue spectrum exactly hits 0 right at the phase transition and then bounces back.
Mm-hmm.
So you can think of, and it turns out, I don't think I put the slide in here, but it turns out that that eigenvalue, the trace of the inverse of this A inverse is also a susceptibility that diverges. So since it goes to 0, the corresponding susceptibility diverges. So it's actually
but okay, so in terms of, like, if I was actually looking at this system, you said,
yep.
Okay, if you change the death rate, you would see diverging fluctuations.
Yes.
In diverging fluctuations of individual populations, or
yes, so species abundances. So what you see
is that average species abundance or individual species abundance?
You would see in average species abundances. What you see is basically what you'd see is that you become extremely sensitive to perturbations around that point. So basically what happens is around this kind of point, all species, yeah, I mean, you just become extremely sensitive. You can either talk about the trace of the matrix, or you can talk about the eigenvectors of the matrix.
Yeah.
It has pretty much all the characterizations of a replica symmetric first or second order phase transition in any disordered systems thing. So it could be like the SK model or the Huffield model, all these kind of things.
Okay.
But I think physically what you want to say is that you just become very sensitive to perturbations, which is kind of the physically interesting stuff. And I don't take the model seriously enough. In some deep, say, on a mathematical level, that's definitely true. But I think the point is that I think rather than whether it's a full phase transition or a crossover, because that seems to depend a lot on the model we choose, I think what's interesting is this kind of you suddenly hit typicality, and it's kind of weird. It's kind of weird, but Wenping found that this was well understood in the last 10, 15 years in information theory, but definitely in the last 15 years.
Thanks.
I have a quick question about the sort of the abundances or universality of communities where you have these nested structures in your scientific report paper. And then you said the only way to break it is to basically constrain the number of species per community. Is it what you said?
No, so I think the way to constrain it is that basically the way we set up the simulations is we have a regional species pool. I should stop trying to I can't see you when I share my screen. That's why I keep stopping to share the screen. But let me go back to this thing and go back. And so the way we set up, let me see if I'm sorry. The way we set up these, the way we usually set up these simulations is that we have some regional pool, which is basically all the species that can exist. And then what we say is we take some fraction of them, right? And we dump them into a community with a resource and some MI chosen, right? The M is the same for all species. You can vary it too, it doesn't matter. But we take a regional species pool. So there's a a potential regional species pool. These are all the species that exist in the universe, and some fraction of them go to each community, and then we see what survives, right? And the point is that you could either have this be a lot of species in each community, or you can turn out that this is very bottlenecked, right? So if we have a very large bottleneck in this immigration step, this random sampling step, right? So that basically you have very few fraction of regional species. What colonizes a community dominates what ends up there, then you get this kind of break this nestedness. That's all we're saying. It's not a very deep statement, except saying that if what's limit is so there's two processes that can limit what species live in a community. They can all immigrate there, and then a lot of them can go extinct because they're competing with each other. Then you're going to get this kind of nested pattern. On the other hand, what could happen is that most of the species don't end up in a community, no matter what community you look at. This is what's called the island effect in ecology often, that it's immigration limited. In that case, you're not going to see this nested pattern.
Okay. But if I just look at the plot, it seems like there's just you can rank your species, and there's just you will have one species that's just robust in all sorts of environment.
Yeah, there's always fitter species and not fitter species, right?
So it doesn't, yeah, but that alone does not explain this.
No, that alone doesn't explain it. Again, I view this as hypothesis generation. Like when I see a large scale pattern, I'm not saying it's the answer. I'm saying that it's kind of unfulfilling that people don't even have mechanistic hypotheses for what might be generating it. And so what I would advocate is this large scale simulation and modern modeling is at least a way of generating testable ecological hypotheses about these patterns.
Again, you somehow asked what is the nested structure?
Sorry, I can't hear you.
Chufan, were you sort of saying, okay, I can see that one of these bars is longer than the others, and there's some kind of scale over, there's some kind of distribution over which their prevalence decays. But were you saying like you kept on referring to a nested structure, and I missed what you meant exactly by that nested structure. I don't know if Chufan was getting it exactly as well.
Yeah, I just meant you sort these things and you get upper triangular stuff. And the point is that I can clearly, I can make the immigration bottlenecks more and more extreme, and I can kill them for sure. And all I will tell you is that that's not evidence of anything, except that I can just assure you that it was very hard to kill this structure. That was the only parameter in our model we could change that consistently killed it. So it generates a hypothesis, right? That this nestedness is pretty easy if you're dominated by competition. It seems like if you're dominated by dispersal, which is what happens on islands, then you shouldn't see this effect. So that would be the test. I would say, okay, the prediction of this model is that if I go just measure stuff that's limited by dispersal, these kind of places where it's hard to get to, and I just focus on those communities, it wouldn't say that. I'm not. The model is good.
But the nested structure. But what you mean by the nested structure? And sorry if I'm repeating this for people who got it. But you mean that if the most fit organism that's prevalent in the most places is basically prevalent everywhere everything else is.
Yes.
And then the fifth most prevalent is present everywhere where the 10th most prevalent is.
Yeah, exactly. So you basically the stuff like the organisms, there's organisms that you only find in the most rich communities, but you're not going to find them in less rich communities. That's what the upper triangle says. Everything that's really fit occurs everywhere. And then you look at stuff. So the stuff that occurs everywhere, there's organisms that occur everywhere, and then you can rank the organisms. And basically what happens is there's not organisms, and then you rank the communities by richness. So basically what you're saying is that I should just
is this ignore, like, do extremophiles just give too small a signal to appear on that? Because like something that lives at 100 degrees in a vent somewhere isn't very prevalent, but the prevalent stuff also isn't occurring in that niche.
Yeah, it's just too small a signal. So you can ask, how did they sample this things? It's kind of random. It was like kind of convenience sampling, It's a big Earth microbiome. These catalogs are or catalogs. I mean, you can.
So there's some.
Those are very small signals. I haven't looked at it carefully, but I would imagine so the scattered plots that, you know, there are little dots that are off the lower triangle. I should go check those before I give this.
But it's probably some statement about the typicality of environments.
Exactly.
On like, as in they're kind of similar, but some are more have more food.
Yeah, that's it. Yeah, that's what we're saying. We're just saying that there's just some harshness of environments.
Yeah.
And everything can get to those same. So and the same basic species can live everywhere. All that changes is the harshness, and that's what's going on. I mean, it's a very dumb thing. I think the main thing I take away from all this stuff is that 16S is not, and these large scale sequencing is not really probing the ecology as deeply as people hoped it would. You know, we're just framing the what I take away is that the experiments we're doing are probing this kind of very coarse structure of ecology, and all the biology and ecology we hoped we were probing with these experiments, you know, is we're not doing it. We need much more refined experimental lenses to see that. That's my take on all this. Not that like the model is amazing. It's just that like if you want to ask about non-typical things, you really have to look closely, not do these large-scale surveys. Those large-scale surveys will just tell you about this, which the physicist in me, as opposed to the biologist in me, is really happy because that's all I care about. I don't have a biologist in me, so I'm happy with that result. But it's clear that it's not answering the kind of questions that most ecologists would be interested in, this kind of data.
I mean, could you also interpret that ranked structure or say hierarchical structure in terms of generalists and specialists? So you have these generalists which can live in any environment, and then you have the specialists which can only live in some environments. In particular, I guess they feed off some waste products of other, maybe some more generalists, or is it something completely different?
Yeah, so you can definitely get it that way, but we don't even need that structure. That's the whole point. We can do it with random Cs.
Yeah, I'm just wondering if one could design essentially a model to, I guess, you know, you asked the question, how can you break it? And you found a way, especially in terms of that referee who hinted to certain things. But could you also design it in set ways that you, I mean, how can you, the question is, how can you design a hierarchy? And I guess you could design it in some way, right? I mean,
I guess so. I mean, I think the question then becomes how much off-target stuff are you allowing, and how does the off-target stuff grow with the thing? And I can just tell you the rule of thumb is that if I'm getting as much energy in any consumer resource model from all the off-target stuff as my primary resource, I suddenly look random. Like, it's related to like a circle theorem and random matrix theory, but I've never been able to prove it. But intuitively, that makes sense, right? If I'm getting energy and the point is that the number of off-target interaction grows. And so the way I assume all this stuff is that it's not at a given time. We know we have diaxic shifts. We know we have lots of stuff. So these models are really meant to be viewed as something that averages over large amounts of time and space. And these kind of consumer preferences represent some average over some large period of time. So over some large environmental point of view, if I'm living off all the other resources as much as I'm living off myself, because say I grow first on sugar, but then I reach stationary phase, you know, well, that's E. coli, but I reach some like non-exponential growth stuff, then I eat all this other stuff. If I'm getting as much energy from the off-target stuff as the primary stuff, it's going to look pretty random. I mean, that's the way I interpret it. But, you know, again, I'm wary of making microscopic interpretations of these models because they're just not good at that.
Yeah. Hey, so there's a question in the chat box. Michael Mastry, he asked a question before as well, or a question before. So he asked, how do you constrain the random matrices? Is it depending on the environment?
So no, the random matrices are usually Ci alpha are basically usually, you can take them uniform, you can take them Gaussian. What's weird is the negative doesn't matter, or you can take them binary. And then the only thing we do is put different amounts of block diagonal structure on them. And the D alpha beta is usually a Dirichlet. It's a Dirichlet matrix, randomly drawn. We just basically and that's that. So you just draw some shape parameters and it's randomly drawn. And again, we put different amounts of block structure on them. So you can play with all this stuff. It's literally like 4 lines of Python code. If you go to this community simulator package that we made, it's really, I promise you, there's a notebook that has all this stuff, and it's super.
Yeah. Okay.
It's super easy to play. We're not there's nothing there's no cheats there, but it's basically very easy, and there's no tricks. It's just basically block diagonal, and actually the thing you draw it from doesn't seem to make much difference. That's the other funny thing. Like for some properties it does, for most properties it doesn't. So I put a link to the paper in the package.
I mean, so when you say negative, it could be also negative events. That means essentially that an influx becomes an outflux. So what does negative in this?
Yeah, negative means that you take some resources that hurt you, and it doesn't seem to matter because of universality. It doesn't seem to matter much if you draw from Gaussians or uniforms or positive or things like that, because as long as the variance of the random matrix elements is finite, you basically are in the same universality class. Of course, it matters in extreme cases. So what does kill you is long tails. As in all random matrix theories, once I get long tails, you get completely, you don't get self-averaging, right? And that makes a big difference, but everything else doesn't seem to make too much difference.
So another quick question. I'm still fascinated by your sort of generalist in what you find just by these random matrices. Did you go back and look at the C matrix for those top ranked species, and is it just the case that the row of their input is just high, just randomly all high, or is more intricate than that?
We checked it. I don't remember. I mean, they're definitely enriched. So if you do like take the top and the bottom, they're definitely fitter. But it wasn't like fitness was an easy explanation, right? Like it's part of the story.
So if you go back and just rank your C matrix, and that ranking wouldn't be exact, or maybe you have to subtract off your leakage matrix.
Yeah, I think the correlation was something like 0.4 or something like that, 0.5. So it was high, but it wasn't everything. No, I don't think there's anything deep going on there. I think it really is your basic intuition with some noise thrown on. There's some stuff that's really fit. It survives everywhere. The harsher the environment, because there's environments that are less rich or harsher, so the fitter organisms are enriched in those environments. It's nothing deep. On the other hand, it was nice to figure out that, like, but I guess what we learned, and maybe we should have understood it from the beginning, is that picture basically assumes competition is dominating community structure and not what gets there. All right, we should have understood that in the beginning. Either we were too dumb and we didn't, but whatever it is, it's a systematic way of maybe even understanding the things you should have already understood, which is basically everything I've ever done in my life.
Good. Okay, then any more questions? I mean, there was a question again by Michael about the constraints that there's, I guess they don't have a biological meaning. I think that's what you're hinting towards, right? It's very robust towards whatever you put in.
Only the energy conservation constraint for the metabolic matrix is what I was going to say. That like you want to conserve energy. As soon as you allow energy to be produced randomly, things go bad.
Okay.
Right.
Good.
Spontaneous generation violates the biology. So as long as you don't arouse spontaneous generation, we're good.
So that means that you wouldn't find potentially a steady state even, yeah? So that might be.
Yeah, sometimes it diverges. It just behaves very differently. You get weird stuff. Yeah, I mean, you get weird stuff. It just doesn't well defined. All our numerics kind of blow up. Like bad stuff starts happening.
Okay, fantastic. It was really great to see you again.
Nice to see you, Robert.
And yeah, so then
in my whole life.
We let you enjoy your first day without teaching. I think you said you just finished your StatMech course.
Yeah.
And yeah, enjoy the rest of the term, and Merry Christmas to everyone. And
I think there's a question, Jordan. You want to ask a question?
I was just wondering whether I could ask a quick one.
Sure.
Perhaps. Thanks again for the talk. I caught one of your. A nice version you gave on related material on YouTube recently, which was really great as well. But it's interesting to hear it in this sort of form here today. So the question that I'm wondering about, which is probably slightly better related to ecosystem models where there is sort of like consumption of certain organisms by other organisms. I'm thinking about like ecosystem collapse, right? So if we remove some organism from a certain environment, it could trigger the sort of collapse of the rest of the food chain, for instance. In these microbial settings, how robust are the sort of steady states to the removal of like one unit from the population? Like how many would you have to remove from the population to say like dramatically alter the steady state that's reached?
Okay. Yeah, that's a great question. So I think in all these systems, Volterra, us, Julio, Guy, all these guys, Michael, Barbier, and everyone who's come to it, I think has come to the same conclusion, though I don't know if it's written down somewhere. The interesting thing is when you assemble an ecosystem and then allow species to go extinct, that's how it's assembled. Actually, the resulting ecosystem is quite stable in any setting, right? Until you hit the marginal stable phase in Volterra, even then you get some big rearrangements. But before that, and certainly in these things, it's quite stable. The places where you get big rearrangements when that happens is near this phase transition to typicality, if you have a structured thing and like that. It's in the Volterra models, it's when you basically break replica symmetry, when you get this marginal stability, that you can get big rearrangements and big fluctuations. But for as long as the diversity is small, the resulting thing is quite stable, more stable than it would be otherwise. And in these consumer resource models, it's always more stable, except for at these phase transitions. And that's because there's basically, you can think of it as the spectrum of the gap of the spectrum of the effective interaction matrix. So basically your stability is, there's a way to think about it in terms of gap. So in physics, often we think of things as being stable if there are big, what are called gaps in spectrums. And the relative spectrum, the effective thing you want to look at is you look at the eigenvalues of your Volterra interaction matrix of the things that survive, right? So I assemble this community, lots of stuff goes extinct, then there's species that survive. Now the point is that the statistical properties of the species that survive are very different from the statistical properties of the things you put in. And this is something that Guy Boonin has been emphasizing a lot, really great papers. And so what happens is now you want to ask, how do I measure the stability of the resulting ecosystem? So what I can do is that, at least in numerics, if I'm doing things, I can say, let me look at the eigenvalue spectrum of the interaction matrices. If I have some eigenvalues that are very close to zero, that actually will result in big rearrangements. But if I have eigenvalues that are very far from zero, so there's a gap, then actually it's quite stable to taking out stuff. There's no zero modes. And so it turns out that until you get a near phase transition, so in Volterra only models, you can get near what's called the marginally stable phase. In our models, you have this typicality. But away from these phase transitions, there's generically gaps. So the self-assembled ecosystems tend to be quite stable. And that's very different from an ecosystem where I say, okay, I put in an ecosystem, I don't let it self-assemble, and I ask if it's stable, which is much more a la me. And they just have very different properties. Because basically the things that aren't fit already go extinct, and that creates this gap. So it's just self-assembling and letting things self-assemble. Systems are more robust, modulo this being close to a phase transition.
Okay, thank you.
I think there's one more question. Let's do it. Jonathan, did you want to ask a question?
Yeah, it's just a kind of follow up from that actually. I just wondered.
Okay, last question to Jonathan.
If like all the conditions you just outlined, if something like in these, let's say you're in the gut, the human gut, and you have like an antibiotic treatment where, maybe like huge numbers or even the majority of the species might go extinct. I was wondering if you'd looked at kind of would you still be in that sort of like situation that you just described where it's still quite stable?
It is. It generally, it's functionally stable for sure. You get some little things of stable things, and function in these models corresponds to weigh each consumer preference by the amount of abundance there is. The interesting thing I would say is where this all breaks down. What is the central assumption of my physics and ecological model? The central assumption in all my analysis is that if I have an ecosystem with, say, large amount of resources and large number of species, let me call that N, this large parameter N, that if I change and remove a resource or species, that's a 1/n effect on the environment, right? It's a 1 over the number of things. Now, I think what goes wrong in disease is precisely the fact that if I remove a species or I add a species, I actually don't get a 1/n effect. And it's largely because you globally modify the environment. So for example, in IBD or IBS, I think what happens is that you add this kind of pathogenic species. If you have the right genetic proclivities, you basically cause large amounts of lymphocyte response. But importantly, it actually completely changes the anaerobic to aerobic character of the environment. So you basically go from an anaerobic to an aerobic thing. And that's an order 1 change by introducing a pathogen because it interacts with your genetic background. And there, all these kind of theories have nothing to say, right? Because they're basically mean field theories that assume this small effect. And in fact, I think that's how we should define healthy versus non-healthy microbiomes and microbial communities. If I remove a species or resource and make a small effect, is the effect order 1/n? Or is it order 1? And if it's order 1, that's basically unhealthy. That's atypical. And atypicality, I think I would associate with disease. That's my dogma. That's my dogmatic way of thinking about it.
Thanks.
Okay. Okay, then I think we conclude the event today. So thank you again, Pankaj, and we hope to catch up in the future, maybe next year sometime.
Yes, thank you so much. And I would love to talk to many of you. I've had a crazy semester, so if some of you wrote me emails, I'm sorry. But I'm free. If you have time, I'm free next week. If you want to continue the discussion, I'm really happy to chat with all of you. And I hope to be in London sometime in the next year. And when I am, hopefully you guys have like 3-4 days you can waste with me, because I'm really missing that during the pandemic, talking to people I don't normally talk to about science, ideally over beer. Anyway, thank you so much for having me.
Okay, Merry Christmas and bye.
Merry Christmas.
Thank you.
Thanks.